Welcome everybody to the June MagX meeting. We are delighted to have Lissa Anderson give the presentation, and she will be introduced and the discussion will be led by Chris Hendrickson. Take it, Chris.
All right, thank you, Laura. Lissa, for those of you who don't know, grew up in Virginia, and following undergraduate work at William and Mary, she got her PhD in 2014 at the University of Virginia, where she developed mass spec methods for intact protein analysis with professors Don Hunt and Jeff Shabanowitz, who are very famous mass spectrometrists. Lissa came to the MagLab as a postdoc in 2015 and was incredibly impressive at pairing with the 21 Tesla ICR instrument to establish a world-leading protein analysis user program. She was hired as a staff scientist in 2016 and promoted to research faculty 2 in 2021. In her spare time, I'm using air quotes, Lissa has, with Dr. David Butcher, spearheaded the MagLab Fair Data Initiative and created a really nice environment for monarch butterflies in her backyard. So Lissa, take it away.
All right, can everyone hear me?
Yes.
And thank you. Can everyone see my slide and my pointer?
Yes.
Perfect. Thanks for the introduction. We'll just go ahead and jump in. So the version of the central dogma of molecular biology that we learn has really far-reaching implications on what the rules of life actually are. So when most of us think back on high school biology, we remember the version that was taught to us, which is typically ascribed to James Watson, which is that DNA makes RNA, makes protein. So to the logical of us, that translates to a single gene produces a single protein. So if there are 20,000 genes, there are 20,000 transcripts, and there are 20,000 proteins. My experience as a protein mass spectrometrist has led me to subscribe far more favorably to Crick's version, which he clarified in Nature about 5 years after Watson, which states that the central dogma deals with the detailed residue by residue transfer of sequence information from one biopolymer molecule to another. Now, sequence information here, ultimately it translates into the structure of the protein, but we're talking about, a code a DNA code that's made of 4 bases, an RNA code that is 4 bases, and a protein code that's rooted in 20 different amino acids. And that doesn't explain all of the nuance that we know exists in biology, but that's essentially what the purpose of DNA is, to be a blueprint for how the protein structure ultimately is translated. And so the dogma deals with the transfer of this sequence information from one molecule to another. And this is the most important part to me. Information cannot be transferred back from protein to either protein or nucleic acid. And that really resonates with me, because in the years since Watson and Crick in the 70s, we've discovered that there are really many sources of protein variation that result in many unique molecular forms or proteoforms. That is a unique molecular form of a protein product of a single gene. Chiefly, these include things like RNA splice variation, as well as post-translational modifications. Now I'm showing 2 here as an example, but a PTM is any modification, a chemical modification that's affected to an amino acid side chain of the protein after it's been translated from RNA into protein. And these modifications regulate the activity of the protein, the localization of the protein, interactions that the protein has with other cellular molecules like other proteins, nucleic acids, lipids, plasma membranes, cofactors. So these PTMs are really the key mechanism by which proteoform diversity is increased. And since protein structure defines function, proteoforms have emerged as a new unit of measurement that is really strongly connected to organismal phenotype, because the truth of the matter is that the central dogma is more closely related to this picture. With every year that passes, we find that the role of DNA and the central dogma for that matter is shrinking. And we know that DNA is only one facet of phenotype. Nearly all of our cells contain the same DNA blueprint, it's all the same genes, yet humans are comprised of 200 distinct cell types. And so what makes a lung tissue cell different than a heart tissue cell? The distinctions lie in which genes are turned on and ultimately what proteoforms are expressed and are operating in the cell. And therefore, approaches that are capable of distinguishing between very closely related proteoforms are in high demand. And so one very promising application of direct proteoform analysis that is sort of a holy grail to the biology field is the cancer protein KRAS. KRAS is a GTPase that is involved in the MAPK signaling pathway, and it's present in really, really low abundance within your cell. And yet it modulates and controls or plays a part in so many different functions of the cell that are important and crucial to life. For example, signal transduction. So these molecules are actually responsible for you knowing that your cells have been touched, or that you're tasting something, or you're smelling something. They're involved in protein biosynthesis. That is, they sort of control what proteins ultimately get expressed in the cell. They're also involved in regulation of cell differentiation and proliferation and movement, and the movement of proteins through the membranes. And unfortunately, this protein, which performs so many functions and is so important to life, is one of the most frequently mutated genes in human cancers, especially pancreatic cancer, colon cancers, and lung cancers. It's differentially spliced into two different isoforms, KRAS4A and KRAS4B. And if you look down here, this little bar represents the protein. You'll see there's 100% sequence identity among those two isoforms of KRAS within the first 85 amino acids. And then you go another 80, and they're still 90% similar. And all of the variation here is in the C terminus. And so that makes it really difficult to probe biomolecularly or selectively target with inhibitors, because you don't necessarily want to wipe out all the KRASs in your cell. You want to wipe out the ones or disrupt the ones that are causing phenotypes of disease. And so this is a really difficult target for drugging with inhibitors. And it's also difficult to study in vivo with typical biochemical methodologies. So the proteoform complexity specifically in KRAS 4B stems from mutations in the primary sequence. Several of those are known to be activating, that is associated with cancer cell proliferation, as well as several post-translational modifications. So if you look on the left here, this is the protein sequence that you see here with all these letters. Up here is the plasma membrane. You can see that the C terminus of the protein is anchored into the plasma membrane here. And all of the green and red residues indicate where a post-translational modification is known to occur on this protein. And everything that you see in blue and in yellow include the mutations or the single nucleotide polymorphisms that are present at 1% of the population that you also have to sort of keep in mind and take into account. So if you take into consideration all of these sources of variation, you know, there are over 24 possible KRAS proteoforms. And a major limitation to our understanding of KRAS is that it's unclear over or which of these over 24,000 possible proteoforms these human cells and tumors are expressing and what makes them different. Sorry, this slide is not moving forward, please. All right, so that was first attempted, mapping the KRAS proteoform landscape in cancer cells and tumors was first attempted, and the results published in 2018 in PNAS. And this was a joint effort between some folks at Northwestern University and the National Cancer Institute. And they were able to identify 11 proteoforms of KRAS from all of the samples that they analyzed and found 2 novel modifications. And this is considered a very giant leap forward in the understanding of KRAS proteoform biology, because we're actually seeing what these molecules look like in vivo and being able to tease apart what the differences in the proteoforms that are expressed in cancer cells versus healthy cells are. So the way that they actually do this is sort of alluded to right here, but this is the meat and bones of what I do every day. So I'm going to go into a little bit more detail on the LCMS side of things. So in order to map the KRAS proteoform landscape, essentially what needs to happen is you have a sample, and the sample is complex. And in order to reduce that complexity so that you can improve the quality of your data, you need to separate the proteins in the sample. We typically use reverse phase chromatography, which separates on the basis of hydrophobicity. And so you have your proteins in your sample. They're put in an auto sampler here in the M class. We perform our reverse phase chromatography. And then as the proteins are being separated and are actually eluting from the end of a column in a droplet form, we apply a high voltage to the tip of the column, and that ionizes our proteins into the inlet of the mass analyzer. So we're directly performing electrospray ionization of our samples as they are coming off of the column and directly analyzing them by mass spectrometry. So we use our mass spectrometer as a detector, but we really get 2 pieces of information anytime we do a LCMS experiment, because we get a chromatogram, which tells us the retention time or the degree of hydrophobicity of the analyte as a function of the % B at which it elutes off the column. But most importantly, what we are collecting as a matter of data in the mass analyzer are mass spectra. So at any given point in this chromatogram, you can look at the mass spectrum that was acquired at that retention time, and that tells you what proteoforms are eluting at that time. You need to do a few more things in order to make that identification, but the first part of this experiment, which is all being done inside of the mass analyzer, by the way, in the gas phase, is you collect your survey scan or your MS1 scan, where your goal is to obtain an accurate intact mass of the proteins as they existed in the samples from which they are derived. And remember that we're doing mass spectrometry, so we're measuring the mass to charge ratio of these analytes, but ultimately the goal is to determine an accurate intact mass. After we've gotten our survey scan or our MS1 scan, we choose precursors and we isolate them in the gas phase inside of the mass spectrometer, and we fragment them. And the goal there is to produce a collection of fragment ions that differ in mass by 1 amino acid. And by looking at the mass differences between consecutive fragment ions, you're able to determine the residue mass of the amino acid in that position And that gives you the sequence of the protein. And it also allows you to site localized post-translational modifications, which is effectively, it allows you to determine the primary structure of the protein. And so the 21 Tesla is state-of-the-art for proteoform analysis in that it achieves the highest resolving power per unit time of any mass analyzer in the world. It also achieves sub-ppm mass measurement accuracy routinely for intact proteins. And very importantly, the charge capacity of the 21 Tesla is approximately 10 times higher than that of commercially available instrumentation. And so that really enables us to get spectra that are very high in signal to noise and very sequence informative by improving our dynamic range. We also have several bells and whistles that we built onto the 21T that improves its ability to perform direct proteoform analysis, like this external multiple storage device, which will allows us to improve signal to noise much faster than traditional spectral averaging. And we also have several different types of dissociation modalities and ion manipulation strategies that offer optimal sequence coverage and signal to noise. And these are really, really important for doing the types of, for doing the experiments like mapping KRAS proteoform landscape, because your success hinges on your ability to identify proteoforms. And to identify proteoforms, you have to have really excellent sequence coverage. And this is the best sequence coverage that they reported in their paper. Don't be scared, it's a lot of letters, but these are just amino acids. The green and the red are post-translational modifications, and each of these flags indicates a fragment ion that has been site localized to that amino acid residue in the sequence. But the point is that this sequence coverage is really poor. It's only 24%. And that's the best that they were able to get of all of these proteoforms, which explains why they only were able to report 11 proteoforms, because they just don't have the sequence coverage to differentiate these very closely related sequences and site localize the PTMs. To do that, you need a really powerful mass analyzer like the 21T. And so we obtained a sample of recombinant KRAS, which was provided by the National Cancer Institute. And when we analyzed this at 21T, our sequence coverage, because of the improvements in the mass analyzer and the sensitivity of the instrumentation, improve our sequence coverage routinely to approximately 90%. And this is achieved in an experiment in a span that takes less than 3 minutes. And so after getting those really promising results, and sort of at around the same time in 2018, I received a UCGP grant that made the ICR facility in the MagLab a satellite facility of the National Resource for Translational and Developmental Proteomics, which is based at Northwestern University, which effectively meant that they were interested in a lot of very high impact biological applications, but they wanted to apply our super amazing instrumentation to those sorts of problems. So very shortly after that PNAS paper came out, we were allowed to analyze the same 14 cell lines that were analyzed in the PNAS paper at 21 Tesla. And when we did that, we were able to increase the number of RAS proteoforms that were identified from 11 to 41. We also discovered 2 novel classes of proteoforms. One of them I'm going to delve pretty deeply into, and it's shown here. This is KRAS 4B that was derived from a lung cancer cell line. When we ran this, I actually identified this within hours of us collecting these data, because it just shouted at me. I was like, what are you? So we observed this new species that I had never seen before that was 321 Daltons away from the most abundant form of KRAS. And the MSMS data, or the tandem mass spectrometry data that I got, provided really concrete evidence that the species was the result of C-terminal cysteine residue clipping, which was likely affected by endogenous proteolysis. This is exciting, kind of because I knew that if that residue was clipped, it also removes prenylation, which is in the form of a farnesylation. That's essentially just a carbon chain, a fatty acid chain. And so when the C-terminal cysteine is cleaved, that lipid is removed. And that's what's responsible for anchoring the KRAS into the plasma membrane, where all these signaling events are mediated and occur. And these proteoforms had not previously been observed in the colorectal cancer cells that were studied earlier. And so they analyzed 34 more colorectal cancer tumors. And it turns out that they found this C-terminally truncated form of KRAS in multiple mutant forms as well as wild type forms in 18 of 34 of the cancer tissues, but in only 3 of the healthy tissue samples. In addition, in some of these samples, 11 of them specifically, the truncated form of the KRAS was twice as abundant as the canonical or the normal wild type form of KRAS. And so this was very intriguing to our collaborators at Northwestern and at the National Cancer Institute. And so they did some functional studies. And is somebody trying to talk? I'm sorry.
Yeah, can you wrap up in the next minute or so?
Yeah. They did some functional studies, and they found that in live cell imaging, that normal KRAS, which here is stained in green, associates exactly in the same place that the membrane dye stains in the live cell imaging. And if you merge those images, you can see that KRAS is localized to the plasma membrane of the cell. When you truncate this, golly willikers, there we go. When you express in these cells this truncated form of KRAS, you see that the KRAS is diffused throughout the cell. It is not localized to the membrane, and so it is not actually associating with the plasma membrane. As a result of that, KRAS interacts with RAF here at the plasma membrane. And due to that interaction, normally you see activation of the MAPK signaling pathway. And in KRAS that expresses this C-terminally truncated form, we see that the proteoform can't interact with the MAPK signaling pathway because it's not localized to the membrane. And so we see a decrease in downstream signaling of ERK2 phosphorylation here compared to the basal levels seen within the controls and the parental cell lines. Since the MAPK activation pathway is associated with cell growth and proliferation, it's possible that this truncated proteoform is part of an anti-proliferative regulatory mechanism, which could pave the way for development of new anti-KRAS therapeutics that bypass the need to disrupt splice variant specific or PTM specific membrane targeting pathways. While there were no clear trends between the expression of wild type and mutant KRAS truncated proteoforms, we are going to do sample more tumors and hopefully establish trends between the cancer stage, subsite, or vital status of patients that express these proteoforms. Finally, it's really important that we determine how the KRAS is generated in vivo, and that's some of the work that is going on at the National Cancer Institute in Frederick, as well as the NCI in Washington and at Northwestern. So with that, I want to thank Laura Green for inviting me to present to you today, and Chris for facilitating the discussion that will happen. And I want to especially call out the ladies in red here who actually contributed data that you see on the slides that I just presented. And with that, I thank you for your attention and I invite questions.
All right, thank you very much. If anybody has questions, please feel free to go ahead, or if you want to type them into the chat, you can do that as well.
I probably have a question that's a little bit basic, but every time I look at this stuff and I've talked with you a little bit, it always seems so complex. And, you know, right even in the beginning, how do you know that there is, you know, there is a post-translational mode and in the genes, and how do you know where they are? Or it's post-transgressional, I guess you call it. And when I see, even in the beginning, I don't see how you identify the parts that have changed. And then my second question is, what were the measurements that the Northwestern group used to get the 11 lines as opposed to the ICR that you were using?
I'll answer the second question first because it's easier. The only real difference between what they did in 2018 and what we did is the difference in the instrument. So at Northwestern, they were using a commercial Q-Exactive HF, which is a high field Orbitrap instrument. And for the experiments here, we analyzed the exact same cell lines. I think that they might have implemented some changes in the IP at that point. The immunoprecipitation, that is, for purifying the RAS proteoforms from the cells and tissues, but it was very soon after this PNAS paper came out that, like, this was published in March, and the experiments, the first experiments that I performed with the recombinant protein were in May. So I don't think there were many other changes. And I'm just gonna go ahead and get a different PowerPoint because I wanna put this to bed. It's really difficult to,
If someone else has a question, that's okay. I just could ask you all day, but. And it's probably beyond the scope of this, but I don't know how the external multi-source source increases the signal to noise so dramatically.
yeah, I guess I need to.
Well, I'll take that one, Laura, and just say that, you know, one of the things you get out of the 21 Tesla magnetic field is that you can pack more ions into the trap and have them behave and give you the signals that you're looking for.
I see.
So Lissa drew a comparison with this commercial instrument. We literally can pack 10 times more ions into the trap and still get the great data. If you put that many into the Orbitrap, well, you can't trap them all, and the signal decays really rapidly because of interactions between the ions. It's just too full of charges.
That makes sense. Thank you.
Okay, so, you know,
101. Yeah.
can you see my slide?
I can.
Okay, so this is an example of what MS2 data looks like, or the fragment ion data. And so what we're doing is this is the spectrum where we generate. I really want to find a different PowerPoint to show this to you.
That works.
That works. All right. So we're taking our protein and we're fragmenting it, and we're looking at these consecutive fragment ions. And so the mass difference, or the M/Z difference between this B2 ion here and this B3 ion is 57 Daltons. And I know that 57 Daltons is the mass of a glycine. Then I look at the next one, there's another 57 Daltons. So that means I have another glycine there. This one is 87 Daltons, so I know it's a serine. If it were 87 Daltons plus the mass of the phosphorylation, then I would know it was a phosphoserine. So we're effectively just taking the protein and fragmenting it along the amide backbone such that we create this mixture collection of ions that represent cleavages along every residue of the backbone so that we can put the sequence of the protein back together forwards and in the reverse direction using the complementary fragment ion pairs that we generate.
That's a really good explanation. Thank you. Do you ever, like the kind of spectrometry that I've done with optical and X-ray and stuff like that, you can actually do something called a Rietveld analysis where you can actually generate a spectrum to match it up. Do you ever do that with this kind of mass spectrometry? Try to actually generate the curve that you expect or try to model the comb that you get?
No, we don't do that because we can't really predict the way that the spectrum is going to look because you don't know the chemical-physical properties or the energies of the bonds and the rates at which they will fragment and it's like not a possibility.
I see.
Chris?
Well, there is, I think in silico methods for determining, what that comb is supposed to look like for a given protein. And then comparing the in silico comb to the comb that we get from the data to see if they match well enough to call it a match. You're basically matching those masses. And I think of it as a ladder with rungs of the ladder with different spacing. And so those types of things are done routinely to help you match unknown proteins to a database of known proteins.
But it's not that you're trying to generate the spectrum, you're just seeing if a fragment exists at that M over Z space or not.
Very interesting. These very, very complex. They did a great job of explaining. I really appreciate it.
Any other questions from the audience?
I have a question. Maybe a little bit more technical.
Go ahead.
Is it known, is the mechanism by which KRAS4B gets truncated in vivo known, or is there any idea about how they might happen?
It is not known. The fact of the matter is that this is the first time this truncation has ever been reported. It's not affected by any of the enzymes that we know are responsible for the C-terminal processing that normally happens, which includes truncation of the last 3 amino acid residues usually. In this case, we're losing 4. I think that in terms of the hypothesis regarding what enzyme it is, I'm looking right. Oh, like an enzyme like STE4 or STE24, which is a metalloprotease, which is known to cleave farnesylated proteins at both the motif that we see and at N-terminal distal sites. So it could be responsible, but we don't know right now.
Okay, thank you. Actually, second quick question. I know you talked mostly about KRAS4B, but you did also mention 4A. Is there any indication that it might also exhibit any kind of truncation? I know there's a lot of sequence dissimilarity there, but still.
So we did successfully probe KRAS4A proteoforms, and we described them in a manuscript that's actually under review right now. They weren't able to detect the KRAS4A proteoforms in the 2018 PNAS paper, but we caught some of them here. But basically what those experiments showed us were that the RNA-seq data does not match what we see from the proteoform IP. Like we're not recovering KRAS4A. at levels that we expect to see it at based on the RNA-seq data. So there's either a problem there or the KRAS 4A is getting effectively just degraded. Like it has a very short life in vivo and it goes and does its thing and then it gets degraded by the proteasome.
Okay, thank you.
Yeah.
Okay, I'm going to check the chat. I don't see anything on the chat. Anybody have any other questions?
Great talk.
If not, then let's all thank Lissa. Great talk.
Thank you, Lissa. That was great.